Hello, my name is Sofia Dvinskikh, and today I will be presenting my poster on isolation of debrominating organisms from sponge tissue done in the summer of 2021 in Dr. Hagglom's lab as a part of Project Super. This project focused on isolating strains of debrominating bacteria from sponge tissue collecting at the Great Barrier Reef in order to identify and metabolically analyze the bacteria capable of dehalogenation. The sponge tissue was used to inoculate anaerobic agar shake tubes that were used for selective growth of organisms capable of dehalogenating 2,6-dibromophenol. Debrominating series conducted by introducing the isolated colony from the shake tubes into liquid media with 2,6-dibromophenol allowed us to observe the growth curve of the organisms while increasing the biomass for future experiments. The results of this work will inform future bioremediation efforts on how to more effectively remove organohalide contaminants from the environment. Halogenated organic compounds, also called organohalides, are some of the most problematic environmental pollutants. Organohalides are produced naturally and anthropogenically. Man-made organohalides threaten the environment due to their toxicity and slow rate of biotic and abiotic breakdown. Only a few organisms are capable of dehalogenating those compounds, and some of those organisms are organohalide-respiring bacteria that are commonly found in sponges, due to the production of sponges of other halogenated compounds as byproducts of their metabolism. Organohalide-respiring bacteria use organohalides as terminal electron acceptors during respiration, resulting in dehalogenation of compounds and therefore reduced toxicity. Analysis of this bacteria is a way to understand how dehalogenation takes place in nature and to learn how it can be used to help the environment. In order to isolate debrominating bacteria, anaerobic enrichment cultures established with sponge tissue previously were used to inoculate anaerobic agar shake tubes containing 2,6-dibromophenol as terminal electron acceptor. After sufficient growth was seen, specific colonies were picked up anaerobically and placed in liquid media for growth and debromination series. The rate of debromination was measured through high-performance liquid chromatography. Figure 1 demonstrates the week 0 and week 4 of the agar shake tube setup. 1A, shown on the left, demonstrates the shake tubes immediately after inoculation with a sponge sample, and the serial dilution from factor of 10 to the second to 10 to the 8th. 1B on the right demonstrates the bacterial growth after the growth period of 4 weeks. The isolated colonies are circled in red and appear dark and round. Figure 2 demonstrates the rate of debromination in the active culture EN2C5S acquired from sponge Enthalobusta collected at the Great Barrier Reef. This sample was collected from the shake tube with the dilution factor of 10 to the 5th. The debrominating activity is shown by the increasing proportion of phenol and dibromophenol in the time points of 2, 3, and 4. This project will be continued as my JCHO thesis, during which isolation of the strain will be completed by dilution to extinction of the strain isolated during the summer. After that, the focus will be on the molecular work to identify the strain active in EN2C5S through RNA PCR and analysis of its metabolic pathways that allow for debrominating activity. We thank Nicole Webster from Australian Institute of Marine Science for providing sponge samples from the Great Barrier Reef. This work was funded in part by a Rutgers Global Grant and the New Jersey Agricultural Experimentation. I would also like to thank Dr. Haggblom and Lauren Hall for training, mentoring, and supporting me throughout the development and continuation of this project. Thank you.